Right, so hello everybody. My name is Diane Newell. I do profess to be somewhat of an expert on zoonoses, and you'll probably find out the basis of that over the next few minutes or so. I'm extremely lucky to live surrounded by a beautiful environment in the Test Valley, and surrounded by my domestic animals and the livestock around me. But I've also had a job for most of my life, which has taken me all over the world and brought me into contact with some wonderful animals and wildlife. And what I'm going to talk about today is not the pleasures and beauty of that, but some of the dangers of being in contact with these animals. So we're talking about zoonoses. Well, what are they? I suspect that most of you over the last 2 years have acquired quite a lot of knowledge about zoonoses. And so I'm really sorry if I'm going to preach to the converted, but I'm going to assume that a lot of you don't know much about it because Steve has professed he doesn't, and start from the beginning. So we humans and our animals, our environment, are home to billions and billions of microscopic organisms, which as a whole we call microbes. And that home we generally call microbiomes. So those microscopic organisms are viruses, bacteria, fungi, and some others that are a bit bigger that we might come across. Most of them are good. They're the ones that make the alcohol that goes into your wine. They're the ones that put the air into your bread. The good ones, the ones we want to keep, and the ones that keep us safe. There are, unfortunately, quite a few bad ones around as well. And the bad ones are the ones that put the mold onto the bread that you suddenly want for your breakfast. And there are some really, really ugly ones, and they're the ones that cause diseases and that we're going to come on to talk about. So these are the bugs that cause infections. Infectious diseases are disorders that are caused by these microbes. Most of them are microbes. Some of them are going to be bigger than microbes, and they're things like parasites, worms, some that you can actually see within your eyesight capabilities. And these are pathogens. So pathogens are organisms that can cause infectious disease. Unfortunately, our animals that surround us also have their own microbiomes, and very often those microbiomes contain infectious pathogens as well. Very, very often, those animals are totally asymptomatic, so they have no symptoms associated with the pathogens that they carry. And what we're going to focus on is diseases that are transmitted directly or indirectly from the animals in our environment. So we're talking about infections that are transmitted from wildlife, from livestock, either directly or through the food, and from pets directly to us. But what we mustn't forget is that we can transmit our bugs back to the animals that surround us as well. And there's plenty of examples of that. I'm going to start with man's best friend. 33% of UK households owned a dog in 2020-21. It sort of went up a little bit during the pandemic, but is beginning to settle down. But that's an awful lot of dogs. And this is Janie. Janie is one of the 7 golden retrievers that I've had. I've got 2 at my feet at the moment. And Janie and my granddaughter Megan absolutely loved each other. But Janie, like all dogs, was very fond of rolling and eating anything that was mucky, messy, and smelly. The smellier, the better. And unlike most other dogs, and certainly all golden retrievers, although you could show him a cold, icy river and he'd go straight in, show him a bath full of suds and sweet smelling, and he ran a mile. So Janie's coat was full of nasties from what he was eating and rolling in. And most of those could cause, or most of the bacteria in there can cause some sort of disease. Bacteria like E. coli cause diarrhea very, very frequently. And I have a PhD student at the moment who is clearly showing that some of the bacteria on the coats and feces and urine of dogs can give you urinary tract infections as well. Dogs carry vectors, ticks, that can carry Lyme's disease. And if you're really unlucky, dogs that bite can give you rabies. Dogs are our oldest companions. The number of years that dogs were domesticated ago, it seems to vary with the papers you read, but somewhere past 11,000 years ago, dogs became domesticated from wolves. There were huge benefits to man for that. They got protection, they got the ability to hunt, and they got the ability to clean up the mess that humans left around. And what's been shown with some very fancy archaeological work now is that dogs and humans shared their microbes, like a whipworm, for example, here, which is a Trichuris vulpus. We've shared those microbes with our canine friends for thousands of years. Gerard Diamond, in his absolutely fantastic book, Guns, Germs and Steel, in 1997, declared that all people on Earth formerly were hunter-gatherers. And recent work from the same sort of group has shown that these hunter-gatherers suffered from lots of infectious diseases, just like other wild primate populations. And there's some really stunning paleoarchaeological and DNA sequencing work from ancient DNA in dental plaques, which now shows many of the diseases that we're quite familiar with, like anthrax, were around in these human populations. But millions of years of evolution gave those ancient hominins mechanisms to fight infectious diseases. And those mechanisms were called immune responses. So when any animal sees a foreign invader, like a pathogen, it responds to it with a development of immunological mechanisms like antibodies, and I'm sure you've heard of T cells lately as well. Ancient hominin DNA from people like Neanderthals has recently shown that the genes associated with immunity were enriched in these ancient hominins. We have actually inherited some of that DNA enrichment in immunity-associated genes. So we did this by a process of transferring genes from these hominins into our European Western type populations by processes that we assume are sexual. So the ancient Neanderthals and the non-Africans, so the humans, the Homo sapiens that rolled out of Africa up through the Middle East and into Europe, actually um, integrated their DNA, and they have joined in and produced a hybrid immunity, which enables us to respond very well when we encounter new pathogens. And we can thank our Neanderthal ancestors for that. About 1 to 3% on average of our DNA is in fact Neanderthal based. My husband's is 4%. I have to think about that very often. The presence or absence of these Neanderthal genes is important to respond to some of these new diseases. And if anybody's been following the COVID-19 epidemic will know that some of these genes, their presence or absence of some of these genes, actually affects whether you get severe symptoms from the viral infection or not. So what's in your DNA really dictates how you're going to respond to some of these severe pathogens. About 11,000 years ago, humans shifted from the hunter-gatherer scenario to a more farming lifestyle. And that began the process of domestication of large mammal species. And we're talking firstly of sheep, pigs and goats, and then the cow, and then the more transport-related horses, donkeys, buffalo, and eventually the llama and the camel. Having these domesticated animals was hugely beneficial to these ancient populations. It provided transport, food, even dung for their fires. It also provided, of course, wealth for trading. But what we clearly know now is that the development of these agricultural communities was associated with the emergence of many human infectious diseases. And many of those diseases were associated with animal origins. Over 60% of the 1,415 species known in 2001 to be human pathogens were zoonotic. To organize or trying to understand how these diseases have evolved and what their outcomes will be has been a constant theme of the last couple of decades. And once again, Gerard Diamond's group has been sentinel in trying to establish what a pathogen will do, what dictates how a pathogen will result in a disease. And they, in 2007, split pathogens into basically 5 stages of disease. These are when they start off, so stage 1 would be when the disease is actually still in the animal. Stage 2 will be when the disease transfers from an animal to a man, and it's primarily still only in the animal as a reservoir, but it can spill over into the man. And then examples of this will be tetanus and foodborne diseases. Stage 3 is when the spillover becomes a bit more prevalent and you get a few cycles of infection in the man, and that we've got examples of that, of plague. Stage 4 is when there are longer outbreaks, so when there are more like epidemics happening and you get transference from one man to another. And examples of that are cholera and influenza. And then finally, we get this 5th stage where it's transferred solely to be a man-to-man transmission. And there we've got at the moment AIDS and measles. COVID-19 sits in the stage 4 classification, but the one that I'm going to start with is actually the foodborne diseases. And this is one that I've been involved with an area for many decades. Most foodborne diseases are derived in temperate zones, and they come from largely from domestic animals. And the main transport route of these diseases is through the contamination of food or water or the environment by animal feces. And we know these diseases have been around for a very long time. And we've been, most public health authorities have been monitoring such diseases because most of these diseases result in diarrhea, and diarrheal diseases have been monitored in the UK since the early part of the last century. Foodborne diseases are a huge problem worldwide, and at the moment it's estimated that there are over 550 million cases, but most of the foodborne disease burden is actually suffered in children under the age of 5. So this is a huge problem for World Health authorities, and because children do not thrive well when they're having repeated episodes of diarrheal diseases due to contaminated food and water. And because of that, there's been a huge amount of research in these areas over the last century, because we can monitor it, we can see it, we know what the outcomes are. And that research has developed in certain areas. And one of them is that it looked at the whole of the farm to fork control and prevention approaches. And the research developed the tools for surveillance and for monitoring the epidemiology of these infections. And huge amount of information has been derived on understanding the pathogen and the host interactions. And a lot of this information, believe it or not, has fed in to understanding what's going on with COVID-19 at the moment. If it hadn't been for all the research of the last 50 years in the areas of zoonotic diseases transmitted by food, we wouldn't have had the models and the approaches that we've had so far in dealing with this disease. There are lots of livestock reservoirs for many pathogens. If you look at this burden of disease graph from Harry Havlar, you can see that the main diseases, foodborne diseases, that have livestock reservoirs, Campylobacter, E. coli, Salmonella, Giardia, etc. These constitute more than half of all the burden of disease we have from foodborne pathogens. The other things like norovirus tend to be human to human transmission, but via food as a route, rather than from a livestock reservoir, although there is growing evidence that some noroviruses have reservoirs in animals. So I'm going to start with my favorite bug, sorry, um, just a little bit about meat production. Most meat is produced from livestock, obviously, although there is some wildlife meat production. Um, Mostly it's chicken, increasingly throughout the world, followed by pork and beef, and then lamb is a very small portion of what we consume. And all of those meat production areas are increasingly intensified. So you'll see here this picture of a meat lot production system in USA, which is very common. Cattle, very young cattle are put into restricted areas, force fed. And that's the same with pigs going into barns and crates, and chickens. Chickens are produced in vast numbers in very small restricted areas. Increasingly, food production, meat production is global, and it's allowed to be global because we can now transport meat very cheaply and very rapidly across the world. Boats with chilled facilities, planes flying in all the time, and improved packaging systems and storage techniques have enabled that cheap transport. Very frequently, our meat now being imported, and increasingly I think this will happen, will come from countries with a lower welfare and safety standards. And these meats are on the retail shelves in your local Tesco's within hours of the death of the animal. And it's increasingly that ability to transport meat very fast in very chilled conditions with good improved packaging absolutely encourages the organisms to stay alive and be there nice and fresh for when you pick them up off the retail shelf a couple of hours later. Pathogens don't need passports. They travel from one country to another quite easily. So I'm now going to talk about my favourite bug, one I spent a lot of time on, and this is Campylobacter jejuni, and its very close relative Campylobacter coli. They look identical. A spiral bacterium, semi-spiral bacterium, has a little projection, a little motor that allows it to travel around through fluids at either end, and it is a very effective pathogen. Campylobacterosis causes over 166 million cases a year. It's fortunately not very virulent, and the death rate, the mortality is relatively low. So those cases will result in about 37,000 deaths. This is a bug that is ubiquitous throughout the world, throughout most animals, all avians and most mammals. It loves nice, warm environments in the gut, and it hates oxygen, the high oxygen of being out in the open air. And it's particularly fond of chicken. It loves chickens. Chicken has the right temperature for it, has the right gut conditions, and it grows there at huge levels. And so the major attributable source of Campylobacter is the handling and consumption of poultry meat. And here we can see the transmission route. So the gut of a single chicken can contain 10 to the 11 organisms, 10 to the 11 Campylobacters. That's Campylobacter pate. And these organisms spill out of the gut during the processing of the carcass. they contaminate the surface and interior of the carcass, and those juices that you splash around your kitchen when you wash your chicken go up the wall, all over the kitchen surface, and over any salad or cooked materials that you've already got. So this is a very easy organism to transmit around, and we have huge problems with trying to control it. It is not possible to control it at the moment by any means other than trying to improve the hygiene of flocks and prevent them picking up the organism in the first place from their environment, and that is extremely difficult to do. About 35% of all the chicken carcasses and pieces that the EU had monitored in the year 2019, which is the last year we have data for, are contaminated with Campylobacter. Probably only about 100 organisms are required to cause you to have an infection. That infection is acute, very painful diarrhea. And if you pick up a small drop of carcass contaminated fluid, you're talking about 1 million organisms in that one drop. So the chance of being infected from a contaminated carcass is pretty high. So having frightened you with that one, we'll talk about another one. Everybody remember this lady? Yes, Edwina Curry, and the killer in your egg. So there we have a very good example of when you change management of livestock, it can result in a new zoonotic disease. So what we have started in 1988 was an epidemic of Salmonella enteritidis of a type called PT4. Don't need to worry what the PT4 is, but I will tell you it's a phage type, but it's just one single type of Salmonella enteritidis. And this organism started to produce large cases of human disease throughout the end of the 1980s and through the 1990s, to a point when the chief medical officer made the recommendation in 1988 that older people should not consume eggs, and young children should not consume eggs. And the reason was because this strain of Salmonella colonized the oviducts of hens, and it then got wrapped up in the egg being produced and contaminated the eggs, and any chicks that were produced from those eggs were also colonized with the organisms, and the eggs that were produced for table eating were contaminated and could cause quite serious illness. This epidemic went on a very long time. There were enormous control processes put in place, and eventually the epidemic was stopped by a combination of flock hygiene controls and vaccination. The vaccination actually was an essential component of stopping this epidemic. The reason this epidemic happened was because farmers tried to stop other Salmonella strains colonizing the chickens. So they improved the life of the chicken, but in doing so, they created an opportunity, a habitat, if you like, in the gut for this new strain of Salmonella to come in and colonize the chickens from the gut it went to the oviducts and then caused the problems that we've seen. So some epidemics are very relatively short-lived and easy to control. Others, not so easy. Chain enable disease emergence, and this one is Brucella melitensis. Its main reservoir is in goats, and we knew that goats, the goats, or it was known that goats carry diseases, but nevertheless they continued to be a major source of food and supplies for ancient populations. But what we didn't realize was that about 7,500 years ago, Central Asians evolved a mutation which allowed adults to consume milk. And before that, if you consumed milk as an adult, you got diarrhea. So the milk consumption suddenly caused an outbreak of brucellosis, which continues to be endemic worldwide to this day. COVID-19 is not the only pandemic ongoing at the moment. Our livestock production, as I've said, is worldwide, and the intensification enhances the disease risk. And a lot of that disease risk is enhanced by people who are unaware of good animal husbandry and often use antimicrobials as a therapeutic or a growth promoter. Antimicrobial resistance is now, especially of foodborne pathogens, is now a serious zoonotic threat. There are nearly 1.3 million deaths worldwide due to antibiotic-resistant organisms in 2019 from a very recent, in fact, last week or so, published Lancet letter. So this is a pandemic that is happening now, and it's ongoing, and we need to be very much aware of. The new and emerging zoonoses are a totally different sort of concept to the foodborne zoonoses. 75% at least of all new and emerging human diseases are zoonotic, and we've known that for quite some time. So COVID-19 should not have been a surprise, and it certainly wasn't a surprise to most of the microbiologists that I know. And we've been warning about it for decades. And in fact, at the end of the 1990s, I developed the network MedVetNet, which eventually became a European network, which her sole focus was to look at zoonotic diseases and how to control emerging epidemics. On average, every 4 months, a new zoonotic disease emerges. Now, that's quite frightening, but I'm afraid that's true. Of course, not all of them are really big, or maybe not all of them are so serious. For example, in Australia back in 2004, Hendra virus was transmitted from horses to humans. Now, the horses got it from flying fox bats that were roosting in a tree that the horses sheltered under. And the horses transmitted it to 7 people, most of whom were veterinarians, and caused 4 deaths. So that was a very small spillover outbreak. That's what we call these outbreaks. We call them these little outbreaks that go in this way. We call them spillovers. And it only caused 4 deaths. But when you think of that, that's 4 deaths out of 7 is a pretty high mortality rate. and you wouldn't want that becoming an epidemic. Bigger epidemics are much more serious. And another spillover happened in 2014, and that was in the country of Guinea in Western Africa, and that was Ebola. Now, Ebola hadn't been seen in Western Africa by that time, but a 4-year-old boy was playing under the tree of the fig tree that free-tailed bats were roosting in. And he was the first case of Ebola in that area. That outbreak of Ebola eventually caused over 1,500 deaths. But of course, we have today COVID-19, which matches anything that we've seen in our lifetimes anyway. And I looked on the Johns Hopkins website this morning, and we're now up to 400 million cases worldwide and 5.7 million deaths that we know about. And you can accept that that's going to be a really big underestimation. And we'll come back to Ebola a little bit later. So what are the risk factors for emerging zoonosis? Well, it's not a random event. Viruses are much more likely to be emerging zoonoses than bacteria or fungi. And viruses and bacteria that have a big host range that can go right across several host species are often more likely to be zoonotic. Often zoonotic diseases are vector-borne, and some of them, obviously, as I've said before, can become transmissible between humans, and that's like COVID-19. That's when you start, and influenza, when you start getting serious, big pandemics. Many of the pathogens evolve because they have an ability to change their inherited material, either DNA or RNA, as in RNA viruses, which COVID-19 is. And some of them, particularly bacteria, are able to acquire DNA from other organisms, from other close relatives. I've already said that domestic animals and foodborne diseases are most likely in temperate zones, but in tropical zones, it's much more likely to be wildlife that is the animal risk. And as you can see here, timing wasn't quite right, but the danger lurks in the tropical regions. The big environmental factors associated with emergence of new zoonotic diseases are the changes in land use, agricultural changes, and travel, and the things like antibiotic development. We have to remember there is a huge interaction between humans, their environment, and the animals within the human environment. So we're interacting all the time, humans with their livestock with perhaps feral animals that are transferring across between wildlife and livestock. Feral pigs comes to mind, for example. This is going on all the time. Percent of the emerging zoonoses that we know about just lately have originated in wildlife. And the rate of that emergence is increasing all the time. So there's more, as we interact more with our wildlife and livestock, we increase the risk of spillover between diseases in the wildlife and disease in humans. Unfortunately, the way we are treating our planet is increasing this risk. As we lose our native forests, and boy, have we lost a lot over the last few years, we lose the buffer zones between the forests and us. For example, 24 people died from rabies, from vampire bats in South Africa in the last 5 years. Also, as we lose our native natural ecosystems, what is left is fragmented, and it tends to be interspersed with crops. And this leads to loss of biodiversity. We now know that loss of biodiversity in our environment is one of the biggest factors, because biodiverse environments allow a dilution of pathogen transfer. So it means pathogens have more hosts between us than between the original reservoir and us humans. And if you don't have that biodiversity, then the risk of a transfer of a pathogen to us increases. And then there's this horrible association with eating exotic animals, bushmeat, etc. The illegal animal trade is the 4th largest global illegal industry. So it is a hugely valuable industry. And bushmeat in particular is valued at hundreds of millions of dollars a year. Wildlife farming is the new industry for a lot of this. And so the wildlife is actually farmed, whether it's for fur or for parts to go into medicines, putative medicines. These farms supply the trade to the wet markets, and the dangers of wet markets are really obvious now. Up until a few years ago, the most obvious outbreaks from wet markets were the flu virus. And in 2015, there were 924 human cases directly associated with wet market contact with live poultry. It was hoped that regulations would reduce wet markets, but then we got COVID-19. And I'm not going to go into much of this, except please don't think of the conspiracy theories. All the evidence from what you've heard today and all the evidence in the science would suggest that this COVID-19 was most likely from direct contact or from an intermediate host, possibly a pangolin, probably in the Wuhan wet market. But COVID-19, or the virus that causes COVID-19, is a really good example of an evolving pathogen. And it's one that we can actually see happening in front of us all the time at the moment. All microbes evolve by random mutations, and natural selection tells us that the best mutants will survive to grow. And those mutations allow survival in the new and changing environments that we humans keep creating for these pathogens. And those mutations allow the organisms to do things like avoid host immunity or antibiotics. And they allow the evolution of novel strains. And we know all about that. We've seen that with COVID-19 as new variants are happening at the moment. Every 4 months, a new variant comes along. And this will not stop. This will continue. And the other thing about COVID-19 is it has shown in front of us, as we have watched it, hopping backwards and forwards between man and man's closely associated animals, either in zoos or pets or pet shops with hamsters just recently, that the organism actually changes when it goes into a new species. And then that change can be brought back into the human population disease as a spillover. So new variants are occurring with these spillovers backwards and forwards between man, animals, and back to man again. This is a report from the World Economic Forum published about a month ago, which says we are not prepared for another pandemic. The global average score in health security is only about 28.4 out of 100. That is not enough to keep us safe from the next pandemic. And what they reinforce this by saying over 100 countries in the World Economic Forum show little or no attention to zoonotic disease. What can we do in the future? We really have got to have One Health approaches. One Health approaches started about 20 years ago, and they involve the global multidisciplinary detection and surveillance systems that are needed to detect new pandemics and respond to them. We need a rapid response capability, which is prepared, so we don't have to scrabble around looking for PPE. We need to reduce contact with wildlife and reduce habitat loss. And we need to prevent biodiversity loss. We need to stop exploiting wildlife and its habitat. And this is not just a Western world problem. This is a developing world problem and a Western world problem. So we need to educate at all levels, governmental, regulatory, and particularly right across the human and the animal medicine sectors. And then there's climate change. I won't go into that because we're running out of time, but anthrax has just turned up in huge cases in Siberia as we lose permafrost, and West Nile virus is sweeping over North America as as temperatures rise towards the north. And mosquitoes are transmitting this from birds back to humans. So in summary then, humans, domestic animals, wildlife and their microbes live in evolving environments and constitute a dynamic and an interconnected ecosystem. Abe Osterhaus in 2016 said, "It's virtually impossible to predict what pathogen or threat will be and where it will come from and when it will strike." And that was shown clearly with COVID-19. We need a One Health approach. We need to reduce and manage the risks. So we need more research. We need to improve biodiversity and save our habitats. And we need to improve awareness at every level, from school children right up to the most important people. And when I told this grey-haired old lady that her dogs probably carried nasty bugs, she wasn't at all impressed. Thank you very much.
That's great, Diane. Thank you very much indeed. Absolutely.
Fantastic.
Fantastic, yes.
Yeah.
Good, Barry.
I'm sorry it got a bit rushed at the end, but
No.
I realised that time was going on and there were probably questions.
Very good.
Okay. Sorry. Thanks. Yeah, I'll kick off just to begin with by once again thanking Diane for such sharing her great wealth of experience and expertise to really give us what I thought was a really thought-provoking and actually quite scary. Look at where we
not to be scary, didn't I?
no, but we need those last few slides about what we need to do really kind of brought it into focus. I mean, I think the thought that 75% of the new diseases are now zoonotic in origin should really focus our minds, and it got me thinking that just how over recent years, decades, we've become so complacent about food hygiene, about agricultural practices. We play fast and loose with all of these things, and then it's only when something like salmonella sort of bursts through into the public realm and we begin to sit up and take note that we can't carry on like this. I've got one question to kick us off with, and it kind of follows on from salmonella. We all remember that from the late 1980s. And then it was in the 1990s, the next big breakthrough thing that got everybody really quite jumpy was of course BSE. That was one of these prion diseases, wasn't it? Which were very mysterious. But I mean, the bottom line from that was bad agricultural practices, feeding animal waste to livestock, and something jumps across. And I just wonder if was BSE actually a zoonotic disease? Because what is a prion? What are these things?
It's a really good question. I can tell you there is still arguments about whether prions are live or not, and they're on the fringe of what biologists define as a living organism. Yes, we did actually call it a zoonosis. It was included. I remember going to a WHO meeting in the States, and we classified all the top at that time, all the top level zoonotic diseases into what level of seriousness they were. And actually BSE was at the top of the list, and yet BSE only ever caused just over 200 deaths. And so the dangers that we saw then were actually exaggerated by our fear of having a disease that would cause neurodegeneration. Nobody wanted that. And so there was an overemphasis on BSE in terms of the scientists had an overemphasis on it. But in fact, it could have been much more serious if the controls had not been put in place. So it was quite a difficult one to deal with. It hasn't died out. There are still pockets of BSE around because, of course, it got transmitted from cows to calves, which no one was really expecting. But it is controlled, and I don't think it's the danger. I think there are more dangerous pathogens around. Does that answer your question?
Yes, that's a great relief. I remember there was all sorts of. I remember John Selwyn Gummer, the minister, feeding his daughter a burger.
I know.
Really?
I was at the animal, what was the Veterinary Laboratories Agency in those days, and what was quite interesting, this may come under the Official Secrets Act, so don't tell anyone, was that some of my close friends actually discovered BSE, and it was the pressure put on the scientists not to actually make a big deal of it. And so there was quite a lot of pressure on some scientists not to publish, not to talk about it, because they didn't want to keep the minister awake at night.
As I recall, BSE was touted as a disease that could, it was a ticking time bomb, essentially, for humans, and it might not prove to be evident for maybe 20 years. And at risk of showing my ignorance, is it possible that you know, the rapid increase in dementia across the world is in some way related to the prion type problem in the brain?
The monitoring of neurodegenerative diseases is quite good. Surveillance across the world is quite good. And there are very distinctive pathological features of BSE that are just very rare. I mean, they're a group of diseases that, so it's not just BSE is now part of a group of similar diseases. So there are other causes, but I don't think BSE is the danger that you might think it is. And we are well past the 20 years now.
Okay.
Phew.
Hey, thank you. Should we just run through the chat box quickly? A few comments there, and then there's quite a number of hands up for you, Diane. So, let's move on. Reen says, it seems a lot of suffering and death could be prevented by promoting plant-based diets and or supporting research for artificial meats.
Okay. Yeah.
I'm seeing a lot about that.
So the plant-based diets, I try, as my husband will attest, I try my hardest to get us to eat as much plant-based material as possible, and I grow my own. You should be aware that a lot of quite considerable outbreaks of foodborne disease have been generated by plant material. And there are various reasons for that. The main reason is a lot of things like fresh salad crops come from developing worlds where the crops are watered with brown water. And that then may not be cleaned enough by decontamination processes before they're packaged. So there have been big outbreaks of E. coli from spinach crops, for example. In Germany, a few years ago, I remember being involved in one of those. So fresh foods like plant crops can be contaminated with fecal material. There are 70 billion animals, livestock animals in the world, and they're producing about twice as much feces as the humans. So that's a lot of fecal material floating around in various environments. And it's not surprising that some portion of it will get onto our food crops. The other thing that is fairly new is that some food crops, and tomatoes and spinach are good examples, actually the bacteria, like some strains of Salmonella, can get inside the leaves and the fruit. So even though you wash them, you don't wash what's inside. So the answer to your question is some crops can be, you need to wash your fruit. If it says wash your vegetables, then wash them.
Okay. Thank you for that. Here's an interesting one from Anne. I buy eggs from a local farm with hens wandering around outside. The eggs are unwashed. How should these be treated? Are they more or less likely to be affected than nice, clean supermarket eggs?
Oh, okay, it's a really good question. So one of the things that the organic people push forward is that organic eggs, for example, are better for you. Organic chickens are better for you because they're outside in the environment. In fact, they carry considerably more Campylobacter and Salmonella uh, than the intensively reared ones. As with all these things, you should look at the risk and try and produce some mediation, some way of mitigating that risk. So if you wash chickens, you're going to wash the pathogens all around your kitchen. Never wash your chicken. Pat it dry, put it in the oven. If you're taking eggs from a local supplier, which I do as well, I don't wash them. But then on the other hand, I would boiling water will kill anything on the surface. If there is obvious wet fecal material, then you'd wipe it off, obviously. But I wouldn't do any more than that. Just make sure that when you crack an egg, you wash your hands afterwards. So, but that's normal. You should be washing your hands all the time in your kitchen and being aware that there are pathogens around every time you handle something from an animal.
Yeah. Thank you, Diane. Good advice. I think we ought to take some live questions now. Peter Weston, you've got your hand up. Would you like to unmute?
Yes, but first of all, I'd like to echo your comments, Stephen Barry. This is a really, really fantastic presentation. It's made me very scared. Obviously, I got scared, and I wonder if you sleep at night. That's not a question. It's just a comment, really. Because anyway, I'm interested in. We use vaccination to protect us, and there's this issue with bovine TB, where they are saying, and we just kill things indiscriminately by the looks of it. And there's this obviously transmission from potentially badgers to cows. There's the issue here that they say they can't tell the difference between the disease and a vaccination in livestock. That's the reason they're not vaccinating livestock. We basically eliminated TB in our populations by vaccination. Can you try and explain to me why it's so difficult to tell the difference between a vaccination and the actual disease, please?
Okay. Oh, this is a difficult one to do without something to draw on. Okay. So the first thing is to say that the TB that we had is not the same as the TB that we're talking about in cattle and badgers. So it's a different organism. And in fact, the one from cattle and badgers is called Mycobacterium bovis. And the one we had is Mycobacterium tuberculosis. Now, Mycobacterium bovis can go into humans and did, especially through milk and from cows that were infected with Mycobacterium bovis. And that is why we basically need to control TB in cattle. It does influence whether we can export beef and milk and things like that anyway as well. So I was, during the 1990s, very much involved. I ran the TB research unit at the Veterinary Research Laboratory. And so I was very much involved in looking at the immunity and vaccination of cattle. And the reason so cattle are diagnosed with TB by something called a skin test. What you do is you inject a tiny amount of Mycobacterium bovis, slightly processed material, under the skin, into the neck. So it's just under the skin. And then with this disease, what will happen is the immune response in an animal that's infected will produce a lump under the skin in response to seeing the Mycobacterium there. What you do is you compare that lump with another lump from a material that is just a normal, it comes from a soil Mycobacterium. So you compare the two. And if the lump from the Mycobacterium bovis injection is bigger by a certain amount, then you say that animal is infected. The animal is culled and it will go for sampling to check that it actually had TB. And you're right, there are problems. This is a very difficult organism to culture, and there are problems, and it takes a very long time to do. So it's, as a test, it's not a great test. So that's why why the diagnosis in cattle is bad. It is true, and it's not doubted by any scientists who've worked in this area, that there is transmission of this organism between cattle and badgers. That's without doubt in my mind. The problem is how to control that. You're right when you say vaccination is possible, but if you vaccinate the cow, then because of the test I've just explained, the animal will show positive in that skin test because you vaccinated it. And so therefore, under international rules, that animal really should be condemned, culled, and got out of the food chain. There are some newer tests around, but there are some doubts about how effective they are. And you may have remembered just lately the problem with the llama. Llamas are really difficult to test for TB, much more difficult than cows because they have a thicker skin. But the newer, more modern tests have been tried on the llama, and they weren't able, as far as I heard from the last reports, to culture the organisms from that animal.
Thank you, Diane.
I'm not sure whether I've answered your question. It's a really difficult area.
Yeah.
Thank you, Diane. And
thank you. I want to say one thing. If I could have your contact details, I'd be very interested to have your contact details if you want to pass them to Steve. But thank you very much for a very comprehensive answer. Thank you.
Thank you, Peter. I've got a traffic jam here, so come in, Neil Allen.
Hello, yes, Diane, that was really excellent, but there was something that I didn't quite understand. You had a slide up showing a crocodile, and you said about acquiring DNA, and I wasn't quite sure what you actually meant by acquiring DNA.
No. So the crocodile was to show that dangers lurked in the tropics. So nothing more than that. The crocodile wasn't acquiring any DNA unless he ate somebody recently. So when I meant, what I was talking about was that some organisms, and bacteria are a really good example of this, they can transfer DNA from one bacterium to another. It happens by a variety of mechanisms, but when they do, they can transfer over things like antibiotic resistance, for example. Does that answer your question? Thank you.
Yeah.
Sorry,
It was that transfer process.
I was worrying about time by the time I got to that point.
Okay,
thank you, Neil. Thank you. Lee Edwards. Come in, sir.
I put a question in the chat, but basically I was asking your opinion on using bacteriophage treatment as an alternative to deal with antibiotic resistance. So that was the sort of, I just wanted to see whether you were are in favour of that or otherwise. The other thing I was going to make, if my memory serves correctly, prions are an abnormally folded protein, and they tend to catalyze other similar proteins to fold abnormally. And so once they get in the system, they can be quite devastating if they get into the brain in particular. And so they're a very, to even call them life is rather strange, because they're literally, they've got no means of reproduction or anything. They just work by catalysis. So it's very, very strange thing even to call an infectious agent, really, but never mind. Anyway, so I'd like your opinion on bacteriophages.
And you're absolutely right. I deal with the prion things, but you're absolutely right, which is why I think most scientists or most microbiologists I know would question whether they are an infectious agent, but they do transmit from one person to another, as in sort of the Kuru diseases, and they do transfer from animals to man. So they are within some definition of zoonoses. Okay, to bacteriophages, fascinating area, and really this is a direction that an awful lot of people are looking at. I've not worked on bacteriophages myself, but I've got a lot of friends that have and are, and my general feeling is that if you keep applying bacteriophages, you actually end up doing the same as you do with antibiotics. You induce resistance.
Okay, thank you very much. Thank you, Lee. Peter from Newcastle. Can you unmute?
All right, thank you.
Thank you.
Again, can I say what a splendid talk. Thoroughly enjoyed it, Diane.
Thank you.
Diane, I lead the climate change group in Newcastle. And yesterday we had a talk on food choices and climate change. And one of the things to consider, we went through historically about food, and then we were looking into the future. We were looking into lab-grown meat, which is produced, I understand, from getting cells from real animals and then going on from there. But one of the potential problems seemed to be that, sorry, I am a vegetarian, by the way, I don't eat meat. But one of the problems is that the meat we eat, we somehow have a protection against problems in it, which is probably some of you covered earlier in your talk. But with this lab-produced meat, they're not sure that it will be sterile. It will not have any protection inbuilt in it. Does that make sense to you?
Probably not. Yes. I mean, sterility for tissue culture, sterility is essential, and you can test for it. It's we do it all the time. If your culture medium is not sterile, it will look cloudy, it will smell horrible, and it will go a yellow color, not nice pink color, or orangey pink color. If you've got bacterial or fungal contamination, if you've got viral contamination, the cells die. So you've got some obvious indicators of whether something is going to be sterile or not. I'm not sure what sort of protections they think are actually in meat. When you get a lump of beef on your plate that's raw, that hasn't been cooked, there's not much that I can think there that is protective, because the cells will be dead anyway, so there's no immune response. There'll be some normal contaminants that would eventually turn the meat because it's got contaminated, and it'll be become bad meat. But you'll smell that and see it, and I can't think what would be a protection as such.
Unfortunately, I can't come out of my screen to go into my talk to check my source, but I had a good source, New Scientist or something.
Okay, well, I absolutely, if you want to email me with a question, we can talk about it another time.
Thank you.
Right. Thank you both. Ronnie James.
Thank you. Thank you. Wonderful lecture. So simply explained that even though I'm not scientifically minded, I could understand most of it. Thank you so much. My question is it being recorded so that I can review it again? And pass it on to other people that haven't had the pleasure of listening to the talk and learning from it.
Indeed, it is being recorded, so in due course it will be available from a link on our website, Ronnie.
And so you will send us an email connection for it.
No, you can go and pick it up from the website.
Okay,
Thank you. You won't get an email.
Okay, thank you.
You'll just have to look out for it under previous meetings.
Thank you.
Okay, Robert Bonatti.
Right, thanks. Once again, I'd like to reiterate that it was an excellent talk, very, very good indeed. I just bring up a couple of a few points. One is, I remember from the salmonella outbreak among the eggs, that what Edwina Curry had said was that egg production in this country was riddled with salmonella. What was reported in the newspapers was eggs in this country are riddled with salmonella. And immediately she had to resign, which was very strange because she always came across as a woman who would, you know, stand in her own corner and fight. And she just didn't. She immediately resigned. Yeah. Second thing that came up, I think it was round about that time, I saw something on the television with some cattle which were being brought into this country from France that were riddled with bovine AIDS. Yeah, so as you looked at them, there was a load of slime pouring out of their noses. And the comment was made, there was no evidence of transfer of bovine AIDS to humans at the time. So the question is, was it a good idea to bring them in the country in the first place? And is there any evidence now of transfer of bovine AIDS to humans? And sorry to monopolize, but lastly, I guess all bacteria are mutating at any given time. But what is it that'll change, say, you know, like a flu into a Spanish flu that'll kill 50 million people? I mean, is there any science behind this? You know, why, you know, one version is like really deadly, but the previous version was hardly deadly at all? Okay.
Oh, lots of lovely questions there. So the first thing about Salmonella enteritis and Edwina Curry, I can't comment about why she decided to resign. That's politics, and don't get me going on politics. At the time so 1988, the surveillance showed that about 1 in 200, 1 in every 200 eggs had Salmonella enteritis in it. So it was probably an accurate statement to have made. And it slowly, it increased and then decreased again. So the worry was real. The danger that the chief medical officer presented as making the recommendation that older people and young children should not be given undercooked eggs. So you can have boiled eggs, you know, hard-boiled eggs, but not undercooked eggs, was real. So that's number one. Number two, bovine AIDS. So all the evidence on human HIV infection, which HIV is the infective agent which causes AIDS. All the evidence is that this was derived from, in fact, chimpanzees in Africa. And so this is a particular type of HIV, HIV-1H, and which resulted in the human infection. I'm totally unaware of anything that suggests that it came from any other source. So whether animals should be imported into the country, it's down to regulations that are set by, in the early 80s, that would have been Ministry of Agriculture, later DEFRA. They are set down by various rules. And at any time, even when we were in the EU, governments had the right to, and on public health grounds, to restrict importation of any animal that was diseased. Okay, and then, what was the final one? Sorry,
what would make a mutation? What makes a mutation particularly
Spanish flu? Yeah. Okay. So what we've got at the moment is a really good example of how viral epidemics, pandemics arise. And so flus are basically moving around, flu viruses, and there are many of them, are moving around between the various populations of animals and man all the time. They're particularly going from pigs to humans, chickens to humans, and various types. And if you remember that their flu has a nomenclature, an N and H nomenclature, and some types are more prevalent in some animals than in others. At the moment, we've got, I think, an H1N5 outbreak going on in wild birds. So some of those will transmit to humans. The next point is, if you remember that diagram I showed, how you go from transmitting from animal to human and then human to human, which is of course what happened in the Spanish flu outbreak. It's the transmissibility between humans that causes the pandemic. It's the transmissibility between humans that's causing the COVID pandemic at the moment. Now, what makes an organism more virulent is determined by the genome that it has. So in the case of COVID, the RNA and the structure of the coat of the virus, the way the virus divides, the way the virus gets into the human cells, the way it's then transmitted from where it originally goes in, which is in the mouth, and to the lungs and to the other organs. All those things are determined by the interaction between the host and the pathogen. And in the case, we don't know what happened in Spanish flu, but we know what happens in other flus. And it could be something as simple as one simple change in one gene and to produce a small change in a protein that enabled it to, instead of infecting high up in the respiratory tract, it infects low down in the respiratory tract, managed to get across the blood barrier into wherever and eventually killed people. Those things are happening all the time. They're happening in COVID. COVID is a really good example of how small changes in the RNA are causing differences in the disease presentation. And it's well worth looking at.
Thank you, Diane. Thank you, Robert. I'll take one more live question. Trevor Jones.
Hello. Thank you very much. That's a very informative talk. I've got a question about biodiversity, which you mentioned, but I didn't quite perhaps appreciate the significance. And I guess there's quite a complex interplay between that subject and what you've been talking about. I wonder if you can attempt to enlighten me a little bit very briefly.
It's a lovely question. It is one of the things that fascinates me, is what the effect of biodiversity loss is going to have on the risk of emergence of zoonotic disease. And I've tried it. This is almost a new area that because people are only just beginning to realise the importance of biodiversity loss.
Yes.
And I think what is happening is, so as you get fragmentation and loss of forest habitats, the animals that within those fragmented areas and within the small portion of forest that's left get more concentrated. As population densities in animals and man go up, then the risk of transmission of infection also goes up. Because there are more people for the pathogen to infect. And of course, man is encroaching on those areas, either by farming or by building houses or by taking their water. And you see these horrible pictures of orangutans coming out of the forest searching for food into areas which are devastated with habitat loss. It's not surprising that as that happens, the animals they're going to be fewer diverse, there's going to be fewer animals of different species. When you get fewer animals of different species, the evidence that it's being accumulating suggests that there's no longer the dilution effect. So a pathogen, when it goes out of one species into another, if there are fewer host species around it for it to go into, then it's more likely to go directly into man.
Okay.
Does that make sense?
Okay, yeah, well, there's a lot to think about there, actually. It's quite a...
Thank you, Trevor.
It's a really new area, and it's worth looking at some of the... There's some lovely papers coming out at the moment.
Oh, I'll look out for those. Thank you very much.
Yeah.
You're welcome.
Well, we've been through a lot of the live questions. There's still quite a number left in the chat box, but it's 12 o'clock already. And Diane, you've been working so hard with our group this morning. It really has been a terrific talk and Q&A session, I must say. Would you like to say anything, Barry?
No, just to echo that, it's been really, really wonderful. And when I said a little bit earlier, it's all a bit scary, actually. I think probably it's actually made it a little bit less scary hearing the wisdom of an expert. So once again, thanks from me as well.
Yeah, thank you. So everybody, would you please give Diane a vote of thanks for a wonderful talk and her answers this morning.